Exocytosis and endocytosis are big, big means of cell signaling. The membrane of the cell is the primary interface between the cell and its environment. Without the cell membrane, or without the proteins on the cell membrane, you won't be able to understand what's happening around the cell. It's like being in a submarine without being able to see outside of it at all. So the proteins on the membrane are key for signaling between cells. There are 5 ways I want you guys to understand about signaling in cells. Now, signaling is important because without signals, your cells would not be able to have coordinated responses. It's like each is on their own. You could have dissolved molecules passing through junctions, direct contact between membrane-bound proteins, paracrine signaling, synaptic signaling, or endocrine signaling. We're going to go through each individually. So first, the local ones. Your cells can signal each other through junctions. Remember desmosomes, gap junctions. You can get molecules from one cell into the next. But they have to be cells that are right next to each other, thus local. So this is direct sending of molecules between the cytosol. Yeah, junctions are where they meet. So you have gap junctions, desmosomes, and tight junctions. Tight junctions are sort of rivets. They hold it together. Those gap junctions allow for cytoplasm to flow evenly and smoothly. Signals can be sent from proteins in the membrane. Membrane to membrane contact. If you put a cell, it's like the cell is putting out little flags, and the cell next to it is close enough to read the flags. So their membranes are interacting. You might have what's called paracrine signaling, which is very, very similar to synaptic signaling. With paracrine signaling, you've got a cell secreting molecules through the membrane. So the Golgi apparatus is packing up these molecules, sending them out to the membrane, and then releasing them. Those molecules can then bind to receptors on the other cells. This is also known as the mediated cell response, receptor-mediated response.
These are the 5 ways for
Cells to signal, yeah. So this doesn't, though, travel very far. It only really affects the cells surrounding that cell. Synaptic signaling is very, very similar to paracrine signaling. The difference is you're using neurotransmitters that are created in the Golgi apparatus. For those of you who have taken psychology before, these are probably familiar. So what will happen is you're going to have neurotransmitters created in the Golgi. They're packed down, and they're sent out to an immediately adjacent cell that will then absorb those transmitters, or those transmitters will bind to the outside of the cell, causing a chain reaction inside that second cell. So those 4 types of communication are local. They do not affect cells away from this originating cell. It's like when you're yelling, you can only yell so far, which is creepy now that I say it, but you know, yeah. Everything up to this is local. Yes, junction, passing cell information directly across cytoplasm. The long-distance signal is endocrine signaling. You have hormones, and people are like, oh, teenagers, their hormones control their life. The thing is, everybody at every age has hormones controlling their life. Hormones are the way your cells signal long distance. How your the pituitary gland signals to the rest of your body. How your hormones move from one part of your body to the other. Now remember, hormones are steroids. They are lipids that are not very water soluble. So if you put them in your bloodstream, they can travel a long way. Following your bloodstream, they can then cause cells in other areas of your body to react. One part of your body may send a signal that says one thing, and then it gets to another cell at a different part of your body altogether, and that will cause a cascade to happen within that cell. That may need to lead to local signaling of the other cells around it. So those are 5 big ways of signaling. Endocrine signaling. Long distance. This is the only long distance one I am concerned about you guys knowing this semester. So that's how we get the signal out. Now we need to have a reaction of the cell to the signal. So you're going to have a reception, transduction, and response. 3 stages. You've got to receive the signal, change the signal a little, and then respond to the signal. So step 1 is reception. You get a signal molecule that a cell recognizes. Maybe it binds to a receptor, and the cell's then able to say, hey, there's a signal and it means something. So you receive the signal. Step 2, you transduce the signal. It's slightly modified, so it causes a chemical reaction to happen. Here we've got a signal coming in and binding to a receptor. So that's reception. Then something's going to happen in here that's going to produce messages inside of the cell. Any of you guys ever been on a phone tree at some point in your life where one person sends out, of course that's not a thing anymore, is it? Because you have a mass. Never mind. Okay. Have you ever gotten a signal from somebody that caused you to do a whole bunch of different actions? I don't know. You got a call from your parents that say you have to babysit your brothers and sisters. If that happens, you get the signal. The signal is, you receive the signal from your parents. That signal was, go take care of your brothers and sisters. That causes several more things to happen. You need to start preparing to do things. Start preparing dinner. Start, I don't know, making sure they don't cry because now you're in charge. All sorts of things. You've got to change the signal into something that the cell understands. And that leads to a final response, what the cell actually does. So you get the signal, reception, transduction, changing the signal into something the cell understands, and then a response from the cell that then could lead to more signals being sent to other cells and more things happening. That's called a signal cascade, but we're not getting into that. Pages worth. 3 slides worth, yeah.